I'm Ben Drapkin. I'm a member of the thoracic oncology group at Massachusetts General Hospital and a postdoctoral fellow in the laboratory of Nicholas Dyson in the Charlestown Navy Yard. I was very honored to be invited to speak today at the Chapman Colloquium about cross-resistance in small cell lung cancer. Small cell lung cancer is a common disease with a grim prognosis that hasn't changed significantly since the 1980s. It is a highly aggressive neuroendocrine malignancy that's almost always disseminated at diagnosis and almost never resected. Belying this grim prognosis, small cell is incredibly sensitive to first-line chemotherapy with cisplatin plus etoposide. So the problem isn't what to treat the patients with up front, it's that they inevitably relapse, and when they relapse, they become much less responsive to not just EP, but to other DNA damaging regimens. This is a particularly difficult topic to study experimentally, and over the last 5 years, in collaboration with Anna Farrago, a thoracic oncologist on the main campus, I've developed a panel of patient-derived xenograft models of small cell lung cancer to address the cross-resistance question. Patient-derived xenograft models are models of cancer that are generated directly from patient tumor material implanted without alteration into heavily immunocompromised mice. And it's thought that these models would be subject to fewer artifacts than models derived from cell lines that have to adapt to growing in sparse conditions in vitro. Because of a collaboration with the Haber Laboratory, who are experts in the isolation of circulating tumor cells, we've been able to derive many new models, 66 in total over the last 5 years, from both biopsies and circulating tumor cells from small cell lung cancer patients. And in particular, the ability to derive models from circulating tumor cells permits us to derive models from patients without an invasive procedure, which is particularly critical for deriving models of post-relapse disease. We've taken this panel of patient-derived xenograft models and assessed its molecular and functional fidelity to the disease in our patients to try to figure out whether or not it would be suitable to study cross-resistance. Genomic comparison of the PDX models with patient tumors showed a high degree of fidelity whether the models were derived from biopsies or from circulating tumor cells, speaking to the molecular fidelity of these models. In addition, a panel of PDXs were treated with both the first-line therapy for small cell lung cancer, platinum plus etoposide, and a new regimen developed at MGH for relapsed small cell lung cancer, olaparib temozolomide, both of which operate by generating DNA damage and inhibiting repair of damage. In both cases, across a large panel of models, they faithfully recapitulated the patient responses to both regimens, suggesting that overall, this panel of models is suitable for studying cross-resistance. So when platinum etoposide response and olaparib temozolomide responses were compared with molecular features in each of these models, a surprising signature for cross-resistance emerged, that of low expression of interferon-stimulated genes. This interferon deficient signature is the subject of active investigation, as it may hold the key for why it is that small cell lung cancer becomes resistant not just to first-line therapy, but to a broad spectrum of DNA damaging therapies. Thanks so much.